Dear students, Module 37 Gene Therapy regarding hemophilia. The main objectives are to know about the types of gene therapy, to understand the different approaches used in gene therapy to treat diseases, to explain the techniques used to carry out the gene therapy, to know about the viral and non-viral vectors used to transfer the gene, to understand the gene therapy for hemophilia. Introduction. Many of the human diseases are inherited diseases. Gene therapy is the method for replacing the defective gene with the normal therapeutic gene. This approach plays an important role in many cases of disease. The gene that is used to replace or introduce into the cell is termed as the transgene. This foreign gene, which produces the protein, is necessary for the function in the defected individual. After the transfer of foreign gene into the cell of an organism, then the organism with the transgene is termed as transgenic. The first use of gene therapy carried out in human in 1990 on a 4-year-old girl named Ashanti De Silva. She was suffering from a severe combined immunodeficiency disease with the defected adenosine deaminase gene. Gene therapy has been carried out in many clinical trials. Treating the disease by replacing the defective gene can give more effect than treating the symptoms, but the process of conducting gene therapy is not easier. The gene therapy can be done not only to replace the defective gene, but also for the suppression of the expression of some genes in the organisms. Human gene therapies are conducted with the guidelines. Specific criteria are necessary to carry out the gene therapy, including the proper characterization of the gene before transferring, effective method for the transfer of gene into the cell, minimizing the risk of therapy to the patient, and the selection of disease which are not possible to treat by other methods. After the fulfillment of all these requirements, the approval of gene therapy will be given by the local and national health committees. More than 5,000 patients have been treated. Hence, in this episode, the following aspects are going to be discussed: gene therapy and the types, approaches in gene therapy, techniques used in gene therapy, gene transfer methods, gene therapy for hemophilia. Gene therapy is carried by placing the therapeutic gene which can perform the function of a missing gene or suppress the expression of other genes responsible for the disease conditions. The therapeutic gene is the stretch of the DNA used to treat the disease. The selection of these genes is made carefully depending on the condition of the disease. This gene therapy shows the promising role in case of the inherited genetic disorder, including cystic fibrosis and muscular dystrophy. Gene therapy can be done by transferring the therapeutic gene to the cell of the body. Based on this, gene therapy can be divided into the following two types: somatic gene therapy, germline gene therapy. Somatic gene therapy is performed in the somatic cells of the body other than the germ cells. Somatic cells are the non-reproductive cells which do not show the inheritance. The normal healthy gene is transferred to the body cell and it will not explain any heritability of the effects of this therapy. This therapy seems safer and conservative. It treats a particular cell and the therapeutic effect will last for a short period. The treated cells may die after a few days and needs further treatment to maintain the effect of the therapy. It can also create problems during the transfer of the therapeutic gene to the target cell. However, this therapy is used in many genetic disorders. Germline gene therapy is performed on the germ cells, which are the eggs or sperms. The defects in the germ cells can pass to the next generation. The mutated gene expression can show the diseased phenotypes in the children. The gene therapy in the germ cells can pass to the children. It can show the permanent effect of the therapy in the successive generations. This therapy can prevent many of the inherited genetic conditions. This therapy can be useful if it is done during the embryonic development, where the therapeutic gene can be transferred during pre-implantation or in vitro fertilization. However, it can show effective results. There are many controversies regarding this germline therapy. Some people think that this therapy is not natural, and others are worried about the consequences in the next generations due to changes done in the germline cells. Hence, germline gene therapy is not using in humans. Instead, it can use to produce transgenic animals. The approaches in gene therapy mainly vary in the introduction of a gene into the target cells. The somatic cell therapy is classified into two following types: ex vivo gene therapy, in vivo gene therapy. Ex vivo gene therapy is carried out by treating the target cells outside the body. Cells are altered and then transplanted into the body. The blood or bone marrow or any tissue is taken from the affected individual and the cells are grown in laboratory condition. Then the therapeutic gene is transferred using the vectors. The virus vectors can be used where the virus with therapeutic gene enter into the cells and insert the gene into the cell's DNA. After the culture, the cells with the therapeutic gene are selected and grown. The transplantation of the cells injected into the body again. One of the classical examples for the ex vivo gene therapy is the treatment of severe combined immunodeficiency disease. This disease was treated by correcting the ADA gene. The ADA gene is transferred into the retrovirus, which is genetically disabled. The retrovirus with the ADA gene is then treated with the T cells, collected from the affected individual. The retrovirus infect T cells without the functional ADA. The incorporation of the ADA in the T cells takes place by the infection of the retrovirus, and those cells are cultured in lab condition. The cultured cell with functional ADA are checked and transplanted into the affected individual. The alteration of the cell is made outside the body. In vivo gene therapy is performed inside the human body. The therapeutic gene or the vector is delivered into the target tissues inside the body. This gene therapy can be one of the options if the culture of the cells in vitro is not possible to get a sufficient number of cells. In vivo gene therapy can be carried by using viral or non-viral vectors. Example, treating cystic fibrosis condition. Cystic fibrosis is the condition of lung injury with the absence of functional protein. In normal condition, the gene cystic fibrosis transmembrane regulator CFTR codes for a functional protein. This transmembrane protein will mainly regulate the movement of chloride ions in and out of the cells, leading to the movement of water inside the tissues of mucus producing organs. The movement of water will keep mucus in a thin flowing condition. In the case of the defected gene, the condition of abnormal function of the protein will not help in the exchange of ions and water movement properly. This will lead to the formation of thick, sticky mucus and block the air passage inside the lungs. In this condition, the in vivo gene therapy method helps in the delivery of therapeutic or functional gene inside the lung cells using the harmless viral adeno associated virus vector with the therapeutic gene. Normal functional CFTR is delivered into the lungs by using a thin tube through the nasal passage. The virus will infect the lung airway cells and insert the normal gene into the cell's DNA. Delivery of the functional gene will code for functional protein and carry out the normal functions. Gene therapy can also be categorized based on the different methods used for the treatment. The main methods used in gene therapy are as follows. 1. Gene augmentation therapy. This is a standard form of gene therapy where the defective gene gets replaced by the normal functional gene. Presence of a defective gene in the disease condition will not code for the functional protein. Hence, this therapy helps in the formation of a functional gene product of the required amount by adding the DNA sequence that contains the functional gene. This gene therapy is more effective if the disorders with the effects are reversible. This type of therapy helps treat disease like cystic fibrosis, where the functional copy of the CFTR gene is introduced to replace the defective gene. In case of liver cancer, the functional p53 gene can be replaced to produce the functional protein. This therapy is used to prevent or suppress the expression of other genes responsible for the development of disease. The high expression of a gene can lead to many diseases by producing the gene products more than the sufficient amount. This therapy mainly works in introducing the gene which can inhibit the other gene expression or interfere with the gene product activity. In the case of cancer, the high expression of the gene will result in the continuous cell division. In this case, this therapy helps to prevent the expression of the gene by introducing the inhibitor gene, so that the product of the inhibitor gene inhibits the expression of the faulty gene. The formation of more number of cells with the defected gene can be prevented. The killing of specific cells. This type of gene therapy can be used to destroy certain cells. It is more suitable for cancer treatment, where the continuous growth of cells can be destroyed by inserting the DNA segment, which facilitates the death of the cells. The gene, which is termed as the suicide gene, and the product of the gene will be toxic. The toxic protein can kill the cell. In another way, the inserted gene will produce the protein which can make the overgrowing cells recognized by the immune system of the body, so that the immune response can kill the cells. It is essential to manage the death of normal cells by inserting the gene which is appropriately targeted to kill only the defected cells. Normal healthy gene can be transferred into the target tissue or cells by different means. The two classes of methods are A. Non-viral vectors and B. Genetically altered viral vectors. Non-viral vectors are the actual DNA sequences or the DNA complex molecules used to deliver the gene of interest for the particular disease condition. Using this method, the DNA can be inserted into the host cell directly without using any viral transfection. Non-viral transfer of the gene can be done by using several methods, including electroporation, sonoporation, magnetoreception, gene gun and lipofection methods. The viral vectors are used by inserting the gene of interest into the genetic material of the virus, which are genetically altered. The therapeutic genes replace the disease causing genes. Viruses usually infect the host by inserting their genetic material into the host cell and replicate inside the cell by using the host machinery. Scientists use this strategy to deliver the therapeutic gene by altering the viral genome. The commonly used viral vectors are retroviruses and adenoviruses. Other viral vectors include the adeno-associated virus, herpes virus, pox virus and alpha virus, etc. Viral vectors are used more often which are made harmless. Usually, the viruses infect the host cell by delivering their genetic material and activities in the pathogenic way. So, by using this characteristics feature of viruses, it was able to genetically modify the genome of viruses by removing the virulent genes. Virulent genes usually code for the pathogenic proteins. The viral genes are replaced with the therapeutic genes for the particular disorder can be done. Then the introduction of viral particles can introduce the therapeutic genes into the target cells. Retroviruses are the first viruses which are used as the vectors for the gene therapy. Using this viral vector, the X-linked severe combined immune deficiency was successfully treated. Hemophilia is a bleeding disorder which can be inherited in an X-linked recessive manner. The mutations in blood clotting factor 8, that is hemophilia A, and 9, that is hemophilia B, result in improper clotting of blood. Both factors are produced in the liver. This is mainly caused by the deficiency in the coagulation factor 8 or 9. The mutation in any of these factors show the abnormal functions of these F8 or F9 genes. The F8 gene instructs to produce coagulation factor 8. F8 gene present on the long arm of the X chromosome XQ28. F9 gene provides instruction to produce the coagulation factor 9. This gene also present on the long arm of the X chromosome XQ27.1. The treatment of this condition by gene therapy was initially started with the gene transfer using the viral vector called recombinant adeno-associated virus. The hemophilia A is found more common compared to hemophilia B. Hemophilia A shows in 80% cases and can affect approximately 1 in 5,000 male births. Hemophilia B is less common and shows the frequency of 1 in 30,000 male births across the world. In the severe cases of this disease, a person can experience the hemorrhages in the joints, soft tissues and muscles. Bleeding can also occur life threatening. The gene therapy was successfully used in the treatment of hemophilia. This has been used for a long period and found success after the series of the experiment using small and large animal models. Now, it can be used successfully for the lasting cure by the in vivo gene transfer using the adeno-associated viral vectors having approximately 4.7 kb and are non-pathogenic in human. Currently, there is a phase 1 and 2 gene therapies for hemophilia. The vector genome is manipulated by replacing AVV coding gene sequences with the gene of interest. This can be done under the control of a tissue specific promoter to yield a recombinant ABV vectors. Generally, the human clotting factor 8 coding sequence is 7 kb in length. It was too large to pack into the viral capsid. By deleting one of the domains out of 3 functional domains of factor 8, this could able to pack into the vector and introduce into the target cell. F9 cDNA at 1.6 kb size was easier to incorporate into the viral genome than F8 cDNA. Hence, the earlier study was done for hemophilia B because of the small size of the F9 gene, even though it shows less prevalence. The immune response by the production of antibodies against the introduced factor 8 was eliminated by immune suppression. To conclude, currently advances in technology have improved diagnostic techniques. Gene therapy can be done for the treatment of hemophilia with different methods, and they can show lasting cure. Thank you.